Okay, so in this video, I'm going to look at biodiversity and what the different levels of biodiversity actually are. So the first thing to note is that the Earth has a huge variety of living things, and that's what we call biodiversity. Bio is life, diversity, the different varieties that you can see. So if we're going to look at this and split it up, there are three levels that you have to know. Habitat biodiversity is the first one. So the habitat biodiversity is the number of different habitats within an area or within an ecosystem. So if you look at my picture here, you can see there are lots of different habitats within that area. Now you need to know examples such as there could be meadows, streams, woodlands, sand dunes. These are all different habitats that could be found within the same area that you would be sampling. So it's important to note that when you talk about that habitat biodiversity, you're talking about within a very specific area. Now, if we look here, I am up on top of a hill and I'm taking a picture of the countryside in the UK. So this was when I went to visit a friend, and you can clearly see that there are different habitat diversities going on. If we look at the farmland down at the lower side of the field, what you can see is there is low diversity in a farmland where you have ploughed fields. If you are growing as a farmer a crop, that crop is most likely going to be one type of crop. Therefore, you have one type of species that is growing within that area. That is going to give you a low habitat biodiversity. However, if we look at the countryside, what you would then see is a high habitat biodiversity. If you are looking at, for example, you can see a river, a woodland, a hedgerow, wild grasslands. Because you have lots of different habitats, remember the habitat biodiversity is the number of different habitats within an area. There would be a high habitat biodiversity in the countryside compared to that farmland where you would have a low habitat biodiversity. Now, species biodiversity is the next level that you can identify this variety. Now, a species, remember, is a group of organisms that are able to interbreed and produce fertile offspring. So do make sure you know your definitions. A population is a group of organisms of the same species who live in the same area at the same time. So if we were looking at three different areas, area A, area B, area C, and we wanted to look at the different invertebrate species that are present in that area, we would do a sampling of the area and we would collect the data. So what we have to then think about to understand species biodiversity, there are two parts to this. The first one is to do with species richness. What is the number of different species living in this particular area? Now, if we look, we can clearly see that there is no millipedes present in area A. However, in area B and C, there are each of the invertebrate species present. Therefore, the species richness of area A is the lowest because it only has 3 invertebrate species present, whereas B and C both have 4. Then we have to look at the species evenness. And the species evenness, what we're talking about here is the relative abundance. So the number of individuals of each species that is living in a community. Now, the definition of a community is a group of populations living and interacting with each other in an area. So the species evenness then, if we were comparing this, we have area A has the lowest species richness. Area B has the greatest species richness and greatest species evenness compared to all three. If we were comparing species area B and C, we would identify that they have the same species richness, but area B has a greater species evenness. So it is important that you do identify and really scrutinize if you are given a table of results and looking at species biodiversity. So what we tend to do is we link the species richness and species evenness together in a mathematical calculation when we look at Simpson's index of diversity. So if we want to identify if the biodiversity present within a habitat is high or low, we would use Simpson's index of diversity, and we would normally round that to 2 significant figures. So here I have another sampled data collection. I have this time 5 individual species, and I can see my number of each of those individuals in the column. So what I need to do is I need to get the total number of individuals. So here you can see the Simpson's index of diversity equation is 1 minus the sum of n, for example 25, divided by the total number, which is 85 squared. So I would do that for each of them. 25 divided by 85 squared plus 28 divided by 85 squared, and so forth. And I would do that and continually do that until I get my number at the end, which is the sum of those 5 added together. I then subtract that away from 1, and that will give me my Simpson's index of diversity number. So in this case, using this example, my Simpson's index of diversity number, my calculated number, is 0.75. Therefore, because it is closer to 1, I can say that there is a high species biodiversity in this area that I have sampled. If the value is closer to 0, that is a low diversity, a low species biodiversity, because you are down towards 0. The closer you get to the 1, that's an infinite diversity, therefore a high species biodiversity. What they tend to do in the questions is they tend to give you a set of examples of 2 or 3 areas. In that case, you want to be using the terms higher, lower, because that is you comparing the results that you have been given. What they could also do is they could ask you to use a Spearman's rank correlation coefficient for data. So what they may do is they may give you a set of data to do with collecting and sampling, and therefore if they do that, they want you to use Spearman's rank correlation coefficient. So in this case, they've done some sampling and they've looked at the distance from a river, and they've looked at the mean leaf hair density of the organisms they've sampled. What they expect you to do is to be able to rank each criterion from the highest first and then on to the lowest. So number 1 is the highest, number 10 is the lowest. If 2 numbers are exactly the same, you need to ensure that you add those 2 together and divide by 2. So for example, if your 3rd and 4th value were exactly the same, you would do 7 divided by 2, which would give you a rank of 3.5. Then you would use the equation for Spearman's rank, and you would then work that out to give you your correlation coefficient value, so my RS value. Now do be aware, if it's not Simpson's index they ask you to do, and it's not Spearman's rank correlation coefficient, they can ask you to do a chi-squared test on this. And if they are going to ask you to do a chi-squared test, what they're expecting you to do is to understand that you will be given an expected frequency and then what to do with that expected frequency. Okay, so the next level is the genetic biodiversity then. So all members of a species share the same genes. We know our chromosomes come in pairs, therefore we know our genes come in pairs. We know, however, that there are many different alleles for the gene. So if we are looking at a gene specifically, the more alleles that are present for that gene, the more genetically diverse that population is. So in that case, what we can do is we can say that if the species have a high genetic diversity, they are more likely going to be able to adapt to any change in the environment. So if there is a change in the climate, or if there's a disease that comes along, if they have more alleles present within the population, they are more genetically diverse, they are more likely going to be able to adapt, because those who survive will be able to pass on their alleles. If they all have the same alleles, then a disease could come along and wipe out the whole population. So to have a high genetic biodiversity means you are more likely to be able to adapt to any changes within that environment. So factors that will increase the number of possible alleles within a population are things like mutations. Mutations are those changes to the DNA-based sequence. The gene flow. If we are talking about interbreeding, if there is migration of a population, any migration in any direction is going to cause alleles to be transferred between one population and another. So this could be where you have isolated organisms which are now brought together, for example, in ex situ conservation, and that then results in a gene flow because of the breeding within the situation that we have. Now, factors that will decrease the number of possible alleles are quite high. So selective breeding. If we only select those with the desirable traits that we're interested in and we breed them together, this is going to reduce the gene pool. By inbreeding, we are reducing the gene pool further and further, and that's going to result in more rare breeds. And the reason that's resulting in more rare breeds is because it's more likely that there are going to be more homozygous recessive traits being seen. Captive breeding programs, obviously, if you've got small population that you are captively breeding, that will decrease the possible alleles. The migration, if we were to bring a species in to try to increase the genetic biodiversity, that can help. But if we're just captively breeding in a program, we've got a small population, that will decrease that number. Artificial cloning, asexual reproduction, mitotic division, genetically identical organisms, therefore decreasing the number of possible alleles. Natural selection is going to decrease the number of possible alleles because over time, only those with the more advantageous alleles within the environment are going to remain. Others will be lost. That gene pool is going to decrease. You have bottlenecking. So genetic bottlenecking is where you have some catastrophic event or a change that has happened. So it could be disease, it could be habitat destruction, it could be environmental change. Whatever it is going to reduce the population number. And as it's reducing the population number, the number of possible alleles will also decrease. You have the founder effect. If there is any type of geographical isolation, a river has separated species, or maybe a new road has been built in an area. If you've got a small population, you've got a small gene pool. And then you have genetic drift, random nature of alleles passed on, therefore the frequency of the alleles is going to vary. So these are factors that will either increase the number of possible alleles, so increase the genetic biodiversity, or decrease the genetic biodiversity. Now, what we tend to do is we tend to look at the polymorphic genes. So polymorphic genes is where there is more than one allele present in a blood group. So in this case, you can have the polymorphic gene. So what we're going to look at now is polymorphism then. So polymorphism is where you have polymorphic genes where there is more than one allele. So if we look at, for example, the blood groups, we know that there is codominance here. There is not complete dominance. There's not just one which is dominant and one which is recessive. There is multiple alleles. So if we have multiple alleles, we therefore say that this is a polymorphic gene. So we have the A allele, we have the B allele, and we have the O allele. So you can either be blood group A, you can be blood group B, you can be blood group O, or you can be blood group AB. So if we have polymorphic genes where there is more than one allele, what we then note is we would look at the proportion of polymorphic gene loci. Now, how we do this is we take the number of polymorphic gene loci and we divide that by the total number of loci. Now, remember, loci means position on the chromosome. So if we are looking at this then, and we note that the greater the polymorphic gene loci, the greater the genetic biodiversity within the population, we can identify, if we are to sequence different individuals, we can identify which of the population has the greater genetic biodiversity. So if, for example, I were to take 4 samples of 4 completely different lion species, and I was to use gel electrophoresis to identify the bands of the genetic polymorphic gene loci, what I would then be able to do is identify, using the equation, what the proportion of polymorphic gene loci is. So in this case, I have got 4 different individual species that were isolated from each other and have evolved to be different species. So therefore, I've been told that I've got 73 polymorphic gene loci out of 1,927. So if I use the equation, I can work out my proportion of polymorphic gene loci. So for each of the individual species, I can see, if I call this one A, B, C, and D, I can see by working out the proportion of polymorphic gene loci that my population B has got a greater polymorphic gene loci, and therefore the greater the genetic biodiversity. So biodiversity can be split into any of those 3 parts. It can be the habitat biodiversity, it can be the species biodiversity, or it can be the genetic biodiversity. If you like these videos, please do like and share and follow me, as I will keep putting more videos up.